dar uma resumida. Doutora Drª, eu mandei o paragrafinho que ela mandou para nós
da apresentação dela, né?
Não deu para falar
mas
nada.
acho que dá para resumir, porque acho que ela trouxe muita coisa aqui.
A Emily? Deixa eu ver. Ah, acho que dá pra ler.
Dá para ler?
Dá pra ler.
Hi, Emily.
Tá fechado, ele falou.
Não, tá aberto.
Ué?
Eu não tenho aqui tudo escrito.
O dela tá
Peraí.
montado.
I think you're on mute.
Ô, Fabinho,
Oi.
você falou que tava fechado.
Não, o nosso eu abri.
Vixe, é muito
nesse
comprido.
minuto.
Hi, okay, now you should hear
Ah,
me.
perfect.
Is that better?
Hi, Emily.
Hi, Emily.
Não tá
Hi.
gravando não, né, Fabinho, ainda? Não tá gravando.
How have you been?
Good, how have you been?
Good, good as well.
Vai gravar no
We've just come.
Mas você não pôs pra gravar.
We've just came back from a trip from the north side of the country.
Okay.
Yeah,
Has it cooled down quite a bit, or is it still quite warm?
Well, actually, it's getting warm and warm because we are heading to the summer, and especially up north, it's really, really hot.
Okay.
Vou ler.
Ah, ok, yeah.
Para cortar é difícil. né?
Fala, Fabinho.
No.
O que o Dani falou agora.
Aí o título do webinar.
Okay.
Okay.
So we can align how we
Não,
are
o
going
problema é que ela
to
não tem o
start
título aqui.
this.
Mas eu tenho
Because
Tá
we
no
are
outro
there as
lugar.
well
Eu
on
vou
the
achar.
YouTube
Eu vou
channel
deixar isso aqui
of the
aqui.
company. So
Okay.
that's gonna be recorded in this
Deixa eu achar aqui.
in that social media. So the idea is first. Do que tem que fazer primeiro?
Ela é o host agora. E ela vai. Já deixa pronto que ela vai compartilhar.
So you're the host. So you can get ready and have your PowerPoint presentation ready, all set.
Okay.
Em 3 minutos antes das 2, eu já vou transformar o vídeo na introdução, que é como a gente estivesse saindo da introdução para começar o vídeo.
Ah, tem um vídeo.
Aí fica só ela.
Aí fica só.
Tem um videozinho no negócio.
Deixa eu devia ter escrito aqui.
The plan is, Emily,
Tem caneta aí?
Dr. Drďa are going to talk about you a little bit, like a short introduction, right, on your bio, and then we are going to give you the host admin as soon as she finishes this introduction, and you can then share the screen and open up your PowerPoint presentation in a PPT mode. Right? É isso, então a doutora vai começar, apresenta, passa para ela.
Yes,
Ela
that
só abre, ela vai ter que compartilhar.
Fabinho, põe o seu título da
So
palestra
you,
aí no seu celular para mim.
Esse é do
You will only need to, as soon as we give you the host admin, as soon as Dr. Drďa ends her talk, I'm going to give you the host, you're going to share the screen and then open up the PowerPoint presentation.
Ela já é o host. Já
Okay,
passei.
fantastic.
Eu já passei o host.
Ah, okay, well, you are already the host, so this is okay. So as soon Dr. Drďa ends this brief introduction and pass to you the word, you can just open up the PowerPoint.
Okay, great.
A partir de agora, nem a gente, nem ela vai falar nada, tá?
Fechou já?
Porque agora tá conectado.
Não, porque daqui a pouco as pessoas entram e vão começar a escutar.
Okay.
Fabinho, você
So
fechou?
in about a minute, people are gonna start joining us. So
Okay.
we will all be on silence during this period, and then Dr. Gretchen will start with your introduction.
Okay, great. Yeah. That sounds good. I think I have everything ready to go. And then time-wise, can you remind me how much time I have? I know I have between 20 and 30 minutes, I think was what we originally discussed. But just so I keep on schedule.
Yeah, são 30 minutos mais ou menos de apresentação, né? Yeah,
Corre lá.
it's 30 minutes or so.
Papel.
It's enough. That's enough. Perfect.
Great. Okay, you're back. Great, great.
Olá, estamos aqui como CITOGEM Biotecnologia, Dr. Alexandre Torquio, nosso diretor de produtos, novos produtos, e o Dr. Daniel Rincon, que é o diretor da parte científica, e eu como CSO, Drª Freire Maia. Como todos sabem, a CITOGEM tem, né, por objetivo sempre trazer novidades e apresentando com as novas tecnologias que existem no mercado. São sempre produtos de ponta e hoje nós temos o prazer de introduzir a vocês o novo parceiro, que é uma empresa americana, Quantabio. Durante a apresentação, vocês terão a oportunidade de ver alguns produtos sendo utilizados no processo de um trabalho de biotecnologia relacionado, portanto, com a biologia molecular. Eu tenho o prazer de introduzir a vocês a Drª Emily Putnam, que ela é atualmente a especialista da Quantabio e vai nos falar sobre acelerando descobertas genômicas com tecnologias da Quantabio. A doutora Emily é formada em Biologia e Química pela Universidade de Harvey Mudd College e ela fez a sua tese também nessa área de Biologia Molecular, trabalhando com RNA de interferência e investigando o potencial gênico da terapia gênica para HIV. E ela já trabalhou em outras empresas, atualmente ela está, portanto, na Quantabio, e nesse momento nós temos então o prazer de ouvi-la. Doutora Emily, feel free to talk to all of us.
Thank you so much. I will go ahead and share my screen now so we can go ahead and take a look at our products. Thank you everybody for coming to attend this webinar this afternoon. I appreciate your attendance. Thank you for that lovely introduction as well. So I am here today to talk about some of our newer technologies from Quantabio, as well as some of our key technologies. Now, of course, we won't be able to cover everything we have today, so I'm just going to focus on a few key ones so that we can introduce you to who we are as a company and where we place a lot of our key focused technologies. So as an introduction to Quantabio, we were originally started in 2001 as Quanta Biosciences. And we were one of the first companies to really start to market and generate cDNA and qPCR products, including one of the first to provide master mixes for cDNA synthesis. Beyond that, we have developed additional technology, including our tough technology, which I will focus heavily today and provide some more context on that. In 2016, we rebranded to Quantabio, which is what you would know us as today. And we've used that to kind of leverage our knowledge of enzymes to move into next generation sequencing and provide a few additional products to really round out our portfolio. And then we are always continuing and striving to develop and launch new products. So just within the last year, we've also launched several new products, including 2 kits for RNA sequencing and a new kit for long-range cDNA synthesis. So we have a very long and storied history. We've been on the market for a long time, and we are constantly trying to provide the next generation of enzymes to help improve your molecular biology research. One of the technologies that we are most well known for is our tough mix technology. This is a unique formulation that overcomes PCR inhibitors, which allows for bypassing exhaustive and expensive sample purification. It allows for users to work directly from crude lysates and skip sample purification. And we've also generated these tough mix technology across a wide range of products to allow for compatibility with a wide range of probe designs and detection chemistries. Now, when we talk about PCR inhibitors, some common ones that come across and are from various sample sources. So things like polysaccharides are found in plants, heme and hemoglobin are strong inhibitors from blood, humic acid from soil and plants, and melanin from skin and hair. So as you can see, quite a wide variety of sample input sources have PCR inhibitors present in them. So this can be a common concern in generating robust and reliable sample data. We've taken our long history of engineering application-specific enzymes and paired it with this optimized tough reagent to really provide superior product performance. What this means is that compared to other methods or reagents, these tough mixes are generated to overcome a wide range of inhibitors. So you can apply these to a vast variety of inputs and applications. Are there comments from the team? We're hearing some feedback. Okay. All right. And together this also means, so even though we have this from dirty samples, these tough mix formulations overcome inhibitors that may be in clean samples as well. So they can be applied with clean samples or with dirty samples, but together this tough mix formulation is a very, very powerful formulation for a wide variety of applications. As you can see here, I'm not going to get into all of these specific products today, but I just wanted to show you how exhaustive our portfolio is for a variety of applications. We have multiple tough mixes for PCR, qPCR, one-step RT-qPCR, and genotyping. Some applications that may be more specific to your group or maybe of interest include 16S metagenomics and COVID-19 detection. I'll talk a little bit about pathogen detection using one-step RT-qPCR a little bit later today. But as you can see, throughout our product lines, we have tough mixes available to a wide variety of applications. And I also wanted to briefly show you a variety of our customers from around the world. As you can see, our products have been implemented in a wide variety of market sectors, including biodefense, agriculture, and public health. You can see here just a small subset of our customer base. So there's a very good chance that no matter what type of molecular biology research you are doing, we have a good fit for a product choice for you. Was there questions? I'm getting feedback from the team that I cited him. Okay. All right, just wanted to make sure. Thank you. So to get started, I'm going to focus a little bit more on PCR to start. So one of our new technologies in particular I'm going to introduce. But what hasn't changed is that the needs that are required for enzyme master mixes to perform well. So no matter which choice you make in the lab, you want enzymes that are fast, inhibitor tolerant, able to overcome GC and AT rich regions, allow for high fidelity. So most enzymes can perform well for just a few of these. You may see more specific products out there on the market, but it's hard to find a master mix that's able to do it all. So what I'd like to introduce is our replica Hi-Fi Tough Mix. This is a PCR master mix that's able to generate high fidelity amplification. It features that tough mix formulation to allow for direct amplification from crude lysates. It is inhibitor tolerant with that tough mix. And it's also a long range enzyme. So you have the ability to do both short and long amplicons with quite a bit of speed. So just to give you some more information on the specifics of this product, the extension time can be as fast as 1 kb per second. The total amplicon length can go up to 40 kb. and the fidelity is roughly 90 times greater than TAC. What you can see here are just a bit of the feedback that we have received from customers around the world. And in general, this product has been very popular and very versatile for a wide range of applications. Now, of course, I won't be able to get into all of those today, but I'd just like to show you just a few of these, a few data sets from this enzyme to really showcase the ability of this enzyme in applications, and then I'll show you a few examples as well. So starting with long-range amplification, what you can see here is this product is well suited not only for short amplification, but long amplification. Typically, when you're looking at enzymes on the market, they're able to do one or the other particularly well, but not be able to do all of this together. So the replica hi-fi is very versatile in that it allows you to do all the way from very short to very long amplicons with no real changes needed to the protocol. So very, very easy to use, very, very versatile for a wide range of needs in the lab. As I mentioned, it's also incredibly fast. So the extension time can be as fast as 1 second for any targets under 1 kb. And as I mentioned, this is compatible with long-range amplification. So it is able to go up to 40 kb, and in that case, it's nearly 10 seconds per kb. So this is, if you're familiar with long-range amplification, this is an incredible speed for a master mix. Just showing how you can save time here. So this is looking at a 2 kb fragment and the recommended cycling times for several products that you may be familiar with, including Q5 and Fusion. So just looking at a 2 kb fragment, Replica is able to amplify in only 23 minutes, whereas the other products take nearly double the time. So this is a savings adjustment of half, allowing you to get faster turnaround time and faster answers from your sample. Of course, this time adjustment increases even more as you scale up to larger amplicons, and I'll show you this in just a moment. As far as the tough mix formulation that I discussed, so I mentioned the common PCR inhibitors are found in a wide variety of samples. So for example, in blood samples, hematin is a very common inhibitor. And what we're looking at here is the ability to amplify with high concentrations of PCR inhibitors. So as you can see here, looking at hematin from blood, Replica is able to vastly outperform other enzymes for handling blood samples. Another common sample type is plant tissue, and Xylan is a very common PCR inhibitor to that. As you can see here, Replica is able to handle a large amount of PCR inhibitors, where the other kits are almost able to handle almost none of those inhibitors. So especially for plant tissues, this is a very great and very robust product. Finally, soil samples are also well known for having a large amount of inhibitor concentration. So just looking here again, Replica outperforms these other enzymes for robust amplification. So this is just a small set of samples that are available for use with Replica. We've tested many more inhibitors as well, and Replica just clearly outperforms in terms of tolerance. So as I mentioned, I'll circle back to some applications with Replica in just a minute, but I also wanted to showcase another set of products that can be quite helpful in this arena. So I'm also going to focus a little bit on our reverse transcription products. And what we have done is we have generated a next generation reverse transcriptase for cDNA synthesis. This new reverse transcriptase is our QScript Ultra RT. And it is an incredibly fast and thermostable enzyme. So as you can see here, we have this in 2 formulations. Our Ultra Super Mix is a master mix format that allows for just a simple addition of RNA. And in 10 minutes, you have cDNA generated from that. This enzyme has also been provided in a flexible priming format, which is our QScript Ultra Flex. And again, here, this is able to do only a 10-minute reaction. So you're able to generate that cDNA very, very quickly. just to look at some data from these kits. So the super mix, again, is a single tube master mix with a 10-minute reaction time. It allows for a wide dynamic range from 0.1 picogram up to 2.5 micrograms. And we see very balanced coverage of transcripts. And it's also, so it does not have that tough mix name, but we have tested it to prove that it is tolerant to a wide range of inhibitors here. So you can see in the data on the right, we have again looked at those very common inhibitors from blood and plant and soil. And we see that the Q-Script Ultra Super Mix has no inhibition as determined by qPCR. This is also a very sensitive enzyme, so especially for groups that may be looking at low copy transcripts for gene expression, the Ultra Super Mix handles these low copies very well. So you're able to get very robust and reliable sensitivity from even small inputs and low copy transcripts. Again, we also have this enzyme packaged in our flexible priming format. And this allows for the ability to choose between different priming options. And this also allows for the ability to do very, very long cDNA synthesis. So using gene-specific priming with the flexible priming format, we are able to amplify targets up to 20 kb. So you can see here we're looking at the SINE1 target in the bottom left, and we're looking at a transcript that's roughly 17.7 kb in length. So this is quite a long transcript, as you can imagine. With the UltraFlex, we're able to get full amplification of that full-length cDNA. So this allows for a better look and unbiased look at the full length of transcripts. And we do see improved performance with FFPE samples as well. So how this all comes together, one application that I'd like to show you a little bit more of is cloning, which is a very, very popular application for these products. So looking for DNA, the traditional cloning workflow for DNA starts with your isolated DNA, then goes into PCR amplification, cleanup, and then into cloning. So here we're going to be looking, since we're starting with DNA, we'll be looking at replica hi-fi tough mix. So looking at E. coli genes, which is a very, very common target for cloning applications, we see here very, very quick and robust and clean amplification of a variety of different sizes of products from E. coli. Now, human genes can be a little bit more tricky because they have a different GC content. But here we can see, again, even with small and large targets, we see very clean, very robust results, allowing for very nice cloning results. And then also looking from very long targets as well, we are able to do the amplification for cloning for 20 kb and 25 kb targets and get very reliable results with no non-specific amplification, which can be a common issue for long-range amplification. I also wanted to show a little bit about cloning starting from RNA. So if you're starting from RNA, of course, you'll need to start with cDNA synthesis. So for this, we would recommend our UltraFlex kit. You can then go into the replica hi-fi for the long-range amplification, do bead cleanup, and this can go into cloning. Now, this is just one example here that we've done looking at a coronavirus RNA sample. This is OC43. It's roughly 30 kb in length. And what we've done is we've broken up the genome into roughly 10 to 12 kb fragments. So we used the UltraFlex kit to do a gene-specific priming to get those very long cDNA transcripts. And then we used replica hi-fi tough mix to generate the amplicons from that. So as you can see here, this generated very, very clean data, and it allows for cloning for various bits of the OC43 genome. These fragments can also go into next generation sequencing to do viral whole genome sequencing. Now, what I really want to mention here again is the speed and processivity of these kits allow for a massive increase in throughput and a reduction of turnaround time. So just looking at this particular project with the UltraFlex kit, the cDNA synthesis was done in 10 minutes. And with the replica hi-fi tough mix, the amplification was completed in just over an hour. So overall, you're able to look at and clone very, very large fragments in roughly an hour and a half time frame. So this massively speeds up production and allows for cloning applications. We've had a variety of applications, as I mentioned, used with this particular method. But this just is one highlight of how these products can really simplify and improve your turnaround. Now I'm going to transition here just a little bit. I want to briefly talk about our real-time qPCR products. So for one-step RT-qPCR is a very popular method for pathogen detection. We have multiple products that allow for probe-based detection. And I do want to briefly mention that all three of these products were very popular during the COVID-19 pandemic. So we offer the QScript XLT, the Ultraplex, and the Virus Tough Mix. You'll notice again that these are all Tough Mix formulations. They've been optimized for different amounts of multiplexing, and the concentrations are slightly different. Of course, working together with your CITOGEM team, we are very happy to choose the right product for any application you may be doing. But to showcase how these have been used recently, I want to talk a little bit about wastewater surveillance. So wastewater surveillance has become very popular post-pandemic, especially for looking at population levels of SARS-CoV-2. And especially for wastewater, sewage samples are very, very rich in contaminants and inhibitors. So in this case, it's very, very critical to use a tough mix or inhibitor-resistant mix for this detection. So one customer application here, so this customer was looking for detection of COVID-19 variants in sewage. They had a 3-plexed assay for detecting the COVID-19, and they were using our QScript XLT one-step mix. So what you can see here is just a quick screenshot of their workflow. So they were collecting quite a bit of sewage water, filtering it, and doing RNA extraction. Then by implementing the QScript XLT one-step, they were able to detect 3 to 4 genome copies per liter of wastewater. So that is an incredibly dilute sample that they were able to find evidence of virus within the wastewater samples. So the robust products provide a deep sense of sensitivity and robustness for amplification. Now, I just want to briefly mention that COVID-19 is not the only thing that's out there, and as far as pathogens, these products have also been used to look at PMMOV and HADV in Australia, which is these are gastroenteritis and respiratory diseases. Again, using the QScript XLT. We've also had these applied for enteric pathogens in wastewater and enteroviruses as well. So the QScript XLT, Ultraplex, and the QScript Virus Tough Mix are very, very popular products for qPCR and very robust and reliable for a variety of inputs. Finally, we don't have a lot of time remaining, but I do just briefly want to touch upon our next generation sequencing portfolio. So again, here with the theme of quality, when you're doing next generation sequencing, the key to getting good data out is to make sure that good libraries are going in. So as far as our next generation sequencing portfolio, we have a full portfolio for DNA sequencing. We have 2 options for library prep. So we have a kit that includes enzymatic fragmentation, which is our Spark Frag Library Prep Kit, as well as a library prep kit that's suitable for already fragmented samples, such as FFPE or cell-free DNA. That would be our Spark DNA Library Prep Kit. We also provide a variety of accessory items for doing library preparation, including adapters, Spark Pure Mag beads, and then a library quantification kit for robust and sensitive quantification of libraries. We have also recently launched our Spark RNA-seq kits, as I mentioned, in the last year. For this, we have 2 different kits. We have our Spark RNA-seq HMR kit, which is a depletion-based method for human, mouse, and rat samples, as well as our Spark mRNA-seq kit for mRNA isolation and library prep. So together we now have a full, complete DNA and RNA library prep solution, and this will continue upon the same theme of improving and speeding up next generation performance. Again, today I won't get into the full workflows for these products. We're hoping to have another webinar coming up soon, so stay tuned for that if this is something of interest. I do also want to briefly mention that in addition to the on-demand support that we are always happy to provide, we also have video workflows that go step by step to show you directly how these library preps should be done in the lab, which allows you to always refer back and see how these kits can be prepared. So together we like to provide as much support as possible, ensuring that you have very fast and very sensitive library preparation. Again, this is just a small subset of all of the products that we have within our portfolio. Things that I didn't touch on today are our sample preparation and our standard qPCR products. But of course, we're always happy to support any questions you may have for other products in our portfolio. And with that, I thank you all for your attention and your time, and I welcome any questions you may have.
Thank you very much, Emily, for our explanation and our presentation. It was wonderful to know about Quantabio and the products and the new solutions that we have to PCR and real-time PCR and NGS and molecular biology technologies. For our customers, I think that's very important to know that we are together and now we have some solutions, new solutions for our problems that we have in our routine, in diagnostic lab, in research lab, and for everybody. Daniel, would you like to say something?
No, I think thank you very much, Emily. It was a pleasure to have you with us in this moment and explaining this new technology. I think it's gonna be a revolution in the lab's workflow, and not only due to inhibitors, but the speed and in a practical sense. So thank you very much.
Yes. First of all, the solutions for problems like PCR inhibitors and everything that we have in our routine here in Brazil, in molecular lab in our country. And I would like to say thank you very much, Emily, e dizer para os nossos ouvintes aqui, né, quem está nos assistindo e nos acompanhando, que nós teremos ainda mais 3 webinários com o pessoal da Quantabio falando de produtos específicos. Agora com soluções específicas para cada uma das linhas dentro das técnicas de biologia molecular. Então, não deixe de seguir a gente nas redes sociais, acompanhe a CITOGEM, onde nós vamos divulgar sempre para vocês as datas, os horários, e esse feedback de vocês também é muito bacana com relação ao que a gente traz de novidades para o nosso cliente, para o nosso mercado e para a metodologia diagnóstica aqui no Brasil. Doutora Drª, com a senhora agora.
Muito obrigada a todos. Continue conosco, como já o doutor Alexandre falou, e esperamos no próximo encontrar com vocês. Emily, thank you very much for your presentation. It was very wonderful. And I am sure that our researchers that are here can do a much better work.
E se alguém tiver alguma dúvida, pode contatar a CITOGEM. Estamos aqui para esclarecer qualquer dúvida sobre qualquer produto. Entre em contato nos nossos canais. Por favor, estamos aqui para atendê-los com o maior carinho, com o maior prazer. Muito obrigado a todos.